spermatogenesis and oogenesis, cellular processes and stages. Spermatogenesis and oogenesis are the processes of gamete formation in males and females, respectively. Both involve complex cellular processes, including meiosis, hormonal regulation, and gamete maturation. These processes ensure that sperm and eggs are produced and mature properly for fertilization and successful reproduction. Spermatogenesis, cellular processes and stages. Spermatogenesis is the process of sperm production in the male testes, specifically within the seminiferous tubules. It begins at puberty and continues throughout a man's life, with millions of sperm produced daily. First, cellular process of spermatogenesis. Spermatogenesis occurs in the seminiferous tubules of the testes and involves several stages, from the division of stem cells to the maturation of sperm. Spermatogonia, diploid stem cells. The process begins with spermatogonia, which are diploid 2N stem cells located at the outermost layer of the seminiferous tubules. Spermatogonia divide by mitosis to produce more spermatogonia, or they differentiate into primary spermatocytes. Primary spermatocytes, diploid, spermatogonia that enter the next phase of spermatogenesis differentiate into primary spermatocytes. These are diploid cells, meaning they contain 46 chromosomes. Meiosis 1, reduction division. The primary spermatocytes undergo meiosis 1, a reductional division in which the chromosome number is halved. This results in two secondary spermatocytes, each containing 23 chromosomes, haploid. Meiosis 2, equational division. Each secondary spermatocyte undergoes meiosis 2, which is similar to mitosis but involves the separation of sister chromatids rather than homologous chromosomes. This results in 4 spermatids, each with 23 chromosomes, haploid. Spermiogenesis, sperm maturation. The spermatids undergo spermiogenesis, a process of maturation where they transform into mature sperm cells. During this phase, spermatids develop a flagellum, tail for motility, shed excess cytoplasm, form a head containing the nucleus which holds the genetic material, develop a midpiece packed with mitochondria to provide energy for the sperm's movement. Mature sperm. The final product of spermatogenesis is 4 mature sperm cells that are capable of fertilizing an egg. Second, hormonal regulation of spermatogenesis. Spermatogenesis is regulated by a delicate balance of hormones, primarily from the hypothalamus, pituitary gland, and testes. Hypothalamic pituitary gonadal axis, HPG axis. The hypothalamus secretes gonadotropin-releasing hormone, GnRH, which stimulates the pituitary gland to release 2 key hormones: follicle-stimulating hormone, FSH. FSH stimulates the Sertoli cells in the seminiferous tubules to support sperm development and facilitate the conversion of spermatids into sperm. Luteinizing hormone, LH. LH stimulates the Leydig cells in the testes to produce testosterone, a hormone that is crucial for the development of sperm and secondary male sexual characteristics. Testosterone. Testosterone is vital for the initiation and maintenance of spermatogenesis. It acts on the Sertoli cells to support the process and ensures proper maturation of sperm. The production of testosterone is regulated by negative feedback to maintain appropriate levels. Third, meiosis in spermatogenesis. Meiosis in spermatogenesis consists of two divisions. Meiosis 1 reduces the chromosome number by half from 46 diploid to 23 haploid, leading to two secondary spermatocytes. Meiosis 2 separates the chromatids, forming 4 haploid spermatids, each with 23 chromosomes. Oogenesis, cellular processes, and stages. Oogenesis is the process of egg oocyte formation in the female ovaries. Unlike spermatogenesis, which is continuous, oogenesis is a discontinuous process that starts during fetal development and completes only after fertilization. First, cellular process of oogenesis. Oogenesis begins during fetal development when oogonia, the precursor cells, differentiate into primary oocytes. The process is halted until puberty, when the menstrual cycle resumes. Oogonia, diploid stem cells. Oogonia are diploid cells, 2N, formed during fetal development. They divide by mitosis and form primary oocytes. Primary oocytes, diploid. Primary oocytes are arrested in prophase 1 of meiosis and remain dormant until puberty. Females are born with a finite number of primary oocytes in their ovaries. No new oocytes are formed after birth. Meiosis Y, primary oocyte to secondary oocyte. At puberty, hormonal changes initiate the resumption of meiosis in one or more primary oocytes during each menstrual cycle. The primary oocyte completes meiosis 1, resulting in 2 cells, a secondary oocyte haploid with most of the cytoplasm, a small polar body haploid, which usually degenerates. Ovulation. The secondary oocyte is released from the ovary during ovulation and is arrested in metaphase 2 of meiosis. This is the stage at which the egg is available for fertilization. Meiosis 2, completion of oogenesis. If the secondary oocyte is fertilized by sperm, it completes meiosis 2. This results in the formation of a mature ovum haploid, which combines with the sperm's genetic material to form a zygote, another polar body that is discarded. Second, hormonal regulation of oogenesis. Oogenesis is regulated by the hormonal interactions between the hypothalamus, pituitary gland, and ovaries. This regulation is key to the menstrual cycle and the release of eggs during ovulation. Hypothalamic pituitary gonadal axis, HPG axis. The hypothalamus secretes GnRH, which stimulates the pituitary gland to release. FSH stimulates the growth and maturation of ovarian follicles and oocytes. LH triggers ovulation and the final maturation of the secondary oocyte. Estrogen and progesterone produced by the ovaries. These hormones regulate the menstrual cycle and prepare the uterus for pregnancy. Estrogen also promotes the development of the ovarian follicle, and progesterone is involved in the preparation of the endometrium for potential implantation of a fertilized egg. Third, meiosis in oogenesis. Meiosis in oogenesis consists of two divisions. Meiosis 1 occurs before birth with the primary oocytes arrested in prophase B. It is completed before ovulation, resulting in one secondary oocyte and a polar body. Meiosis 2 occurs after fertilization. The secondary oocyte completes meiosis 2, resulting in the formation of the ovum and a second polar body. Conclusion: spermatogenesis and oogenesis are essential processes for the formation of functional gametes necessary for sexual reproduction. Both processes involve meiosis, hormonal regulation, and maturation of gametes to ensure that each sex cell is capable of combining with the opposite sex's gamete during fertilization. While spermatogenesis results in the continuous production of sperm, oogenesis is more complex and cyclical, involving the maturation of one egg per menstrual cycle. Understanding these processes is crucial for the study of fertility, genetic inheritance, and reproductive health.